My name is Frederick Ruberg from Boston University, the Amyloidosis Center, and it's my pleasure to speak with you for the next 10 minutes or so on ensuring adequate racial and ethnic representation in amyloidosis clinical trials. So just to review so we're all on the same page, we know that amyloid is a protein and amyloidosis is the disease that results from misfolded protein. This figure here illustrates the process by which a protein is synthesized and then goes through different folding steps to ultimately result in amyloid fibrils. These amyloid fibrils are misfolded proteins that deposit in organs and cause those organs to malfunction. There are over 30 or 35 different proteins that can misfold to cause amyloid, and we're going to focus on two of them, but principally one of them in this brief presentation. And the nomenclature is A for amyloid followed by the protein that misfolds. This is a light microscopy specimen showing a heart biopsy with heart cells that are the purple here with the nuclei, and all of this purple proteinaceous material outside those cells, which represents amyloid fibrils. Now, this is not a Congo red stain where the staining would be red, just to make sure everybody's aware of that. This stain illustrates the amyloid deposits in between cells that cause the heart to become thicker and stiffer, and the deposits themselves, as well as the protein intermediates, cause the cells to be injured, die, and lead to organ dysfunction. So cardiac amyloidosis is obviously a very important problem. In ATTR amyloidosis, and we're going to focus on ATTR because that's the disease that disproportionately afflicts communities of color. ATTR amyloidosis results from a protein called TTR or prealbumin that is synthesized by the liver and released into the circulation that exists as a tetramer, and then through conditions that we'll speak about in a few moments, dissociates, misfolds, and then collects in the heart as amyloid fibrils causing restrictive cardiomyopathy, and also in the nerves causing a length-dependent sensory and autonomic polyneuropathy. TTR misfolds into ATTR because of two principal reasons. One is when there is a genetic variant in the TTR protein. That is an inherited condition that causes the protein to be abnormal, that facilitates misfolding, and we call that hereditary ATTR or ATTR-V for variant. Now, ATTR amyloidosis also occurs in the context of no genetic variants, and we call that wild-type ATTR, and we really don't know why this happens. Certainly there are many hypotheses and lots of investigation, including my own lab group. We know that it's associated with aging. We know that it's associated with being male, but we don't really understand the mechanisms fully by why that cause misfolding of ATTR. We do know that in hereditary amyloidosis, the presence of the variant is an important component to that. This slide just really re-illustrates the process that everybody is aware of DNA to RNA to protein through transcription and translation. And the variants occur here at the DNA level. Now, I mention this because the variants in ATTR amyloidosis are passed on in an autosomal dominant manner. And that means the presence of the variant is enough to lead to the manifestations of the disease. And this kindred illustrates an affected father and an unaffected mother having 4 children. 2 of these children have the variant, and 2 of them don't. So that means 50% of the of a affected parent's children or their siblings of the same parentage could have the same variant. And this is important because this helps one understand why the disease is passed on to so many different people potentially, or certainly the variants are. And so identifying a parent and family members becomes really important. Genotyping family members is an area that we don't do very well on, particularly in certain populations, and again, the subject of my personal research. ATTR amyloidosis, owing to the V142I variant, also known as V122I, disproportionately afflicts communities of color in the United States because the variant originated in West Africa. This map of the continent shows the allele frequency of V122I in different countries and different samples. And you can see that the allele frequency is considerably higher in West African countries compared to other countries in the continent. And owing to the reprehensible abduction of people from West Africa and transport to the United States, or at the burgeoning the Americas and then the United States, V122I is a variant that occurs in the US at a very high frequency. In fact, up to 3.4% of African Americans, or 1.5 million people, carry this variant. So it becomes critically important to understand who does and who doesn't have this variant, their risk for disease, and furthermore, because there are now treatments available, to make sure that patients with this variant and amyloidosis are included in clinical trials to test new therapies. We know that heart failure disproportionately afflicts communities of color in the United States. We know that it will cost a tremendous amount to the U.S. healthcare system and affect a large number of people over the next 7 years or so. Unfortunately, persistent disparities have been observed looking at race and ethnicity compared across the population when we're looking at heart failure and heart failure outcomes, such that people who identify as Black have a 2.5 increased risk of hospitalization or death from heart failure irrespective of age. And you can see that that incidence actually is increasing, particularly in this blue curve here, which represents Black males, younger and older people over the past few years. And so we hypothesized, my colleagues and I, that could ATTR amyloidosis, unrecognized, be contributing to the development of heart failure? And again, that is the subject of my research, and it's critically important that we study this question because of the large number of people that are affected. Now, I mentioned clinical trials have been undertaken to test new agents in ATTR amyloidosis, and all of these agents have been approved for ATTR amyloidosis. This agent, tafamidis, has been approved for cardiac amyloidosis. And upcoming approvals will likely come for some of these other agents based upon emerging trial data. But you can see here the proportion of patients who identify as Black as participants is relatively low, very low in these silencer trials. These drugs silence the production of TTR, and still rather low in the tafamidis trial, although not as low as the silencer trials. Now, I would point out these trials specifically enrolled patients with hereditary variants that are not as commonly reflected in the Black population. But it speaks to the fact that these agents may not necessarily be translatable to patients who have different variants, and that's something that we really need to try to understand. How do we make this better? Well, these next few slides speak to the clinical trials in general, not specifically for amyloidosis, but critically important for amyloidosis clinical trials because of the disproportionately high percentage of patients with V122I in the US Black population. This scientific practice statement from the American Society of Preventive Cardiology speaks to some of the problems and solutions that could be undertaken. The problems involve underrepresented and underreported disease, both by sex and gender and by diversity in clinical trials. And this limits the generalizability of treatments, therapeutics, devices to the broad appropriate variation that we see in the US population, the world population, I'd say. And some of the barriers we'll talk about, a lot of it this speaks to the fact of kind of a fundamental lack of trust between caregivers and study teams and the population from which they're trying to draw participants. And these two graphics here, this one focusing on women and this one focusing on diverse populations, provide some suggestions about how these problems could be overcome. They involve avoidance of hidden costs and fully covering costs to participants. Education that also occurs in the context of cultural literacy and sensitivity, and particularly in the case of ATTR amyloidosis. Creating sites that diversely reflect the population of people in the United States, not only in certain regions, having diverse representation in research staff and investigators, and importantly, the principle of community engagement. Now, community engagement I'd like to accentuate as a means to bring clinical trials to participants as opposed to bringing participants to clinical trials. The National Academy of Medicine recognizes this, and in this graphic, outlines the key principles, core principles of community engagement that strengthen alliances between community organizations and healthcare providing organizations and clinical research organizations, expanding knowledge, improving health, and ultimately creating thriving communities with the best possible health outcomes. This nice paper recently published in PLOS One illustrates granular suggestions as to how access, awareness, structural racism, and diversity of the workforce can be improved to improve clinical trial participation. Specific building into budgets of clinical trials and design, transportation vouchers, providing work relief, providing language and translation services, health literacy improvement, educating patients, involving translators to translate trial documentation and provide into languages that people can speak to, understand, and use primarily. Naming and addressing structural racism, training training providers and research teams in implicit bias to recognize and eliminate potential barriers to participation. And finally, creating diversity of the workforce, both in clinical teams and in the investigators themselves, such that the investigative teams reflect the populations that they are drawing participants from, which we know engenders improved trust and engagement. And by doing this, we can begin to address these social determinants of health that we talk about that negatively impact health well-being and outcomes. We, as physician providers, can improve in education. We can create culturally sensitive and meet patients at their level of understanding to bring their level of understanding up. We can engage with communities and with community organizations to communicate the importance of participation in clinical trials for adequate representation so that new therapeutics can be tested. And we also can provide high quality care to everyone and remove barriers that eliminate access to that care. And our organizations and policymakers can help by improving the neighborhood, the built environment, and other aspects like food insecurity and economic instability that negatively impact health and well-being. So with that, I'd like to thank you for your attention.